Chen, you are a second-time awardee of our Birch-Derchin Scholarship, and I think they would appreciate knowing, as would all of us, what the investigative support has meant to you.
The Birch-Derchin Scholarship has helped me tremendously in the last 2.5 years. Inside the lab, it directly helped me to buy the reagents and equipment that I needed for my research. And this research is to figure out how the brain differs between males and females on a biological level. As a molecular biologist and a cellular developmental biologist, I do look at a lot of genes that exist in all of the animals,
yes.
And these are the conserved genes. They exist in pretty much all animal species, me included.
Now you work on the lowly worm.
Yes.
Tell us why you chose the worm as your subject.
The worm is a very interesting organism. The worm has a really complicated nervous system that we can use a lot of tools, including molecular tools, genetic tools, and cellular tools, to sort of understand on a biological level where certain genes are expressed on a single cell level. If we try to see how these genes are regulated on a genetic level, they're quite different. What I found is that these genes exist in both sexes, but during sexual maturation, only the male animals start to express these genes. And that means a whole lot because we know that the female worm and male worm actually do have a lot of different behaviors. And the behavior I am interested in is actually locomotion. You use a video recorder to record how the worms move on a little petri dish for a long time, and you quantify that behavior. You can actually find there are a lot of movement differences between the males and the females. I essentially was able to find these group of genes are expressed only in one sex, which is the male, at a certain time point during their development. If you deplete this gene from the genome that get expressed during sexual maturation only in the males suddenly just become not as active anymore. So this means this gene is really important for contributing to at least part of these sexually different behaviors. In the first 2.5 years using the scholarship, I was able to answer the first 2 sub-questions of my grant proposal. Where are these genes expressed? And the second question is, when are they expressed? Now it comes to really exciting time to figure out what are these genes doing to contribute to the sexually different behaviors, and how are they doing what they're doing? And even more importantly, how are these genes regulated? So the next step will be to deplete some of these genes that showed sex-specific expression earlier to see what happens. That's the basis of why we can use a little worm to understand the biology or biological mechanisms behind all these sexually different behaviors to give us insight into what higher organisms, including ourselves, behave.